Ok. Hola, ¿qué tal? Buenas tardes a todos. Bienvenidos a nuestro seminario del día de hoy. Y bueno, antes de iniciar el seminario, el doctor Luis Cárdenas se disculpa, él es el anfitrión en este seminario. Me pidió que presentara a nuestro invitado, un invitado que pues ya tengo el gusto de conocer. Nos conocemos hace algunos años, ¿verdad, Mauro? Pero no recordaba yo, de pronto la memoria no me llega, ya ha estado de hecho aquí en nuestros seminarios. Me decía Mauro que estuvo hace 5 o 6 años también aquí con nosotros en los seminarios institucionales. Y bueno, es un gusto introducir de manera muy breve la trayectoria del doctor Mauro. Realmente es una trayectoria muy completa. Voy a tratar de hacer un resumen de sus principales contribuciones. El doctor Mauro, él es investigador actualmente del Centro de Ciencias Genómicas. Desde el 2016, hizo previamente un sabático 2015-2016 en el Centro de Ciencias Genómicas, y desde el 16 ya es investigador en el Centro de Ciencias Genómicas. Es obviamente miembro del Sistema Nacional de Investigadores, nivel 2, con una trayectoria muy completa, tiene 130 publicaciones en artículos revisadas, con un total de 10,000 citas, un índice H de 52, y tengo aquí una lista muy larga de las instituciones donde ha venido trabajando. Me explicaba él que inclusive ha estado en la industria, en un laboratorio de polímeros que en Reino Unido, ¿verdad? Entonces, bueno, no voy a dedicar más tiempo. Está igual, con mucho gusto podemos enviarle todas estas contribuciones del doctor Mauro, y sin más agradecerle. Mauro, muchas gracias por estar con nosotros en nuestro seminario, muy amable.
Muchas gracias. Entonces, buenas tardes. ¿Se puede bajar un poquito la luz? Poquito. Ok. No. Muchas gracias, Carlos. Muchas gracias, Luis en familia, por invitarme a esta segunda vez que hago un seminario institucional aquí al IBT. Es un gran placer. And that's the end of my Spanish, and I will start speaking English all the time, of course, with a certain accent, but let me just tell you how happy actually to be here. In a way, it's a very particular year for me. It's the most productive year in a long career for a long time. At the same time, I reached probably the most important paper of my life at the end of a project which is 25 years old. How many people here got 25 years of age? You see, it's a very common type of the year. So the title, it's a form of a tale, una historia. It's so much easier to do it, to organize a seminar as a tale when you are towards the end of a career and when you finish a big project like 25 years old project. And the project, it was basically a quest, like the quest of the Holy Grail. My Holy Grail was the bacterial origin of mitochondria. And this is the only slide in Spanish for you. It's come from my friend, almost a brother. We know each other for over 40 years, Diego González Alfen, del Instituto de Fisiología Cellular. So we know mitochondria, they are the power plants of our cells. The bioenergetic function is a fundamental one. There are many other aspects of mitochondrial physiology and cell biology nowadays are very important, but we will touch them only briefly. Also because, well, the origin of mitochondria is associated with bacteria that had a very similar oxidative phosphorylation respiratory chain. You know, in a way, I'm happy that Carlos didn't go too much into detail in my long international career, but the story of this project is all revolves around the Pacific Ocean. It started in 1998, 25 years ago, like most of your age, when I was at Monash University, and then it was developed further when I went visiting Institute was at the forefront of apoptosis because mitochondria became very important in apoptosis that particular period. And then it's not shown here, but I spent about a decade back to Europe, UK, in Italy, and then I decided to move to Mexico thanks to an invitation of Esperanza, and here I've been still working here. But of a collaboration at the side of the Pacific Ocean with Raquel Quattrini from Santiago de Chile. And to make the importance of Pacific Ocean even more, I will talk you about a bacterial group or lineage called Iodimonadales that have been discovered in Japan. I'm pretty sure there will be some Iodimonadales also here around Mexico, but we shall see later the eventual importance of this. And this is the beginning of my little project. So I like to take notes in these notebooks, etc. And this drawing, which is exactly 25 years ago, my notebook number 3 of apoptosis. This is a drawing taken from a book, a figure in a book of Lynn Margulis. What was really striking, and that's what that writing was about, is that in that period was coming out in the literature that mitochondrial membranes, before I say that, I am the walking encyclopedia of mitochondria because I've been working mitochondria all my life. So I was very surprised, I was also excited that in 1998, cytochrome C comes out of mitochondria and becomes the signal of apoptosis. So I became very interested in all this kind of signaling of cell death, in which it was already established that mitochondrial membranes are attacked by cytosolic factors. One of these is BID, BH3 domain protein of the BCL2 family. And the other thing I predicted just from sequence analysis, and then later I demonstrated, experimented, that BID binds to cardiolipin metabolites, lipids, in particular lysolipids, and therefore BID might be an antibacterial agent, anti-mitochondrial, ultrabacterial. And so the idea just was born in my mind that this type of signaling of cell death attacking mitochondria, it was like awakening the memory of the old bacterial parasite. And I call this project protaptosis, the proto-apoptosis project. And then for over 10 years, the University of Manchester have been working on cardiolipin and BID and apoptosis, and then now there is a question for you here. How many of you know what cardiolipin is? Okay. Cardiolipin is a typical bacterial lipid. It's dimeric. Instead of having 2 acylcysteine, it's got 4. And it is also present in eukaryotes, and very interestingly, only in mitochondria. It's a lipid signature of mitochondria. It's made in the inner membrane of mitochondrion, then it is matured with a complex cycle. And this involved in autoimmune conditions. Originally, cardiolipin was found as one of the components of the lupus erythematosus, so for autoimmune diseases, then in cell death, and now, which has become much more hot topic in the world, mitophagy, the autophagic specific pathway to get rid of excess of bad mitochondria. And you might say, well, is that important? Well, Alzheimer, Parkinson's disease, diabetes, all are to do with dysfunction of mitophagy. So it's a very important process, and cardiolipin is a kind of receptor to initiate the process. In the context of my project, cardiolipin has been one of the first or the most clear biochemical signature, a vestigial relic of the bacterial past of mitochondria. Then from Manchester, I moved back to Italy, and when I was in Italy, I got excited again by this project that I was leaving aside, and then I published with some colleagues who are real microbiologists from Milan, this little paper in which I compared the bioenergetic systems of bacteria and mitochondria, and then for a number of combinations. This paper showed that almost all previously proposed bacterial ancestors of mitochondria can be, and maybe methanotrophs or members of the family Rhodospirillacea, the family of Rhodospirillum, like Tistrella, might be among the living ancestors of mitochondria. That was the idea then, and then the things have matured. In between, when I came to Mexico, I started to collaborate with Esperanza, etc. I even published a book with this title, which is basically the book about the project I've been cultivating, but I didn't know the end of it even in the last chapter, which called Alpha Proteobacteria to Mitochondria. I was putting forward the idea that maybe Rhodospirillacea or some of them would be. And then more recently, from 1921, a complete breakthrough, an unexpected finding that produced a new hypothesis in which marine alpha mags, so you probably know, metagenome assembled genomes related to this Japanese bacterium called Ididamonas might be close to protomitochondria. You will see later why. And one way of doing that was just looking at the aerobic traits which are common between alpha protobacteria, which is a bacterial lineage considered to be closest to the ancestor of mitochondria, and to mitochondria. So the idea is to look in very much detail at this, and this produced, now the title here is because it was a presentation. Believe it or not, we have so many Italian scientists in Mexico. We have our own association, which I was one of the founder, of course, for question of age. And my friend Diego Gonzalez, despite his surname, his mother is Italian, so I can speak Italian, and he presented on my behalf this title, Vado in Mexico, because there's a famous song of a rock star which says, Vado in Mexico. Think of it, and I didn't, it was not the reason I came to Mexico, but it helped to keep me happy to be in Mexico. And this is a presentation of this latest paper with collaboration with a certain Otto Geiger and other colleagues I can see here, including IBT, Alejandro, in which we put together all this work done in many years in a palatable way, so palatable that we were able to overtake the most noxious, the most powerful mitochondrial phylogenetics in the world, and I got it published to Science Advance last August. Before that, I need to introduce the concept, well, mitochondria takes into place at this time during the evolution of Earth, which starts here when 2.3 billion years ago, when oxygen became stable. And half a billion years later, the process is called eukaryogenesis, which we know now it was an alpha protobacterium that somehow encountered an archaean of the Asgard phylum, and together they merged, we don't know exactly how, and the Asgard became the nucleus, and the alpha protobacterium became the mitochondria of eukaryotic cells. And from eukaryotic cells, everything happened. And this process of eukaryogenesis, it can be simplified. Again, I take advantage of my friend's drawing. It's the basis, I lost, excuse me, the base bacteria and archaea join together, they produce the protists, and then plant, fungi, and animalia. This, I know, is the symbols that Diego decided to use for alpha. Asgard, I just ask him why, but it's interesting drawing. In between the book I showed you before and what I did most recently in Science Advance, I got involved into the origin of cytochrome oxidase. It's one of the major respiratory chain enzyme of the, it's the reason we breathe. We breathe because a certain enzyme, which is called cytochrome oxidase, in our mitochondria, use oxygen to oxidize, reduce cytochrome C, and in this produce energy and give us, 1/3 of the ATP. It turns out that the origin of the cytochrome oxidase of our mitochondria comes from this class of bacteria, normally or previously part of the gamma proteobacteria, which are bacteria that live by oxidizing iron. Aciditiobacillia, and in collaboration with the group Raquel Catrini in Chile, to give an idea, these bacteria are used in the largest bioreactor you can think. They are 250, half a kilometer long, 30, 40 meter large. They are used to recycle the copper, which comes as a byproduct of oxidation of pyrites in the tails of the big mines in Chile. So 25% of copper from Chile is made by Aciditiobacillus. That's for biotechnology application, that's a pretty good one. And in Mexico so far, not much. Anyway, so the origin of cytochrome oxidase, which is this enzyme here, is coming from this bacteria. But here you have the other component of the respiratory chain, which reduces cytochrome C, which is a partner that puts together a respiratory chain. Here you have a problem here to move down. Anyway, what I would like you to just to consider that whereas the structure here is a dimer, transmembrane, of the cytochrome oxidase of mitochondria is very similar to the one in bacteria, the structure of the cytochrome C reductase BC1 complex is much more complex in eukaryotes, in mitochondria, in particular because have two proteins here, which made this big domain, which are called core proteins. They are core proteins as a classical name, but they actually are relatives or MPPs, matrix proteins, proteases, which lives in the mitochondrial matrix, are able to recognize the proteins made in the nucleus coming in and cleave off the signal peptide and then move it to the thing, to the rest of the mitochondria. What is very interesting is that plant mitochondrial BC1 complex is the two MPPs, which is a dimer, is part of the complex, whereas in mammalians, in yeast, etc., these core proteins are specific for the enzyme, and the MPP dimer is free, so it's due to a duplication in the system. And one of the most dramatic things for when you compare the respiratory chain of bacteria and mitochondria, there are 3 genes, all catalytic for the structure of COX in the mitochondria. But there is only one, cytochrome B, the membrane, for the BC1 complex. All the others must come later. And here comes, this is figure 1 of the paper we just published in Science Advance. And here is the string, the operon. It actually produces an operon. This is the original one of the iron oxidizing. This is called Acidiferobacter. They have COX2, 1, 3, and other genes. These genes here are used for the assembly because this particular enzyme contains copper and IMA, which is specific only for this enzyme. And then from this original scheme, you have another one, becomes more complicated. Some other proteins are coming in from somewhere, and it becomes a more complicated, sophisticated operon, and then from this are still gamma proteobacteria. Some are lost, some others are coming in, and this is a string with other CTAG and SURF are, again, the assembly protein, and then it comes, and this I found in June 2021. This sequence here, COX2, 1, the rest of the operon, finishing with SURF1. Then there is this M16B. This is a bacterial homologue of the core protein, MPP protein, so the BC1 complex, and then attached to it, there is an operon of the BC1 complex, PET A, B, and C. So this is a kind of a dream screen for somebody like me that was looking for the connection between BC1 complex, which is only one gene in mitochondria, and the cytochrome oxidase. And it turns out that there are some genes and possibly proteins which are the mixture of the two IS-alpha protein NMC in Onchocerca, which is a filarial, a nematode. And some other components found in Tetrahymena COX4, so, which is COX in various genomes. And this combination here was found and still only found in IODD monas and related MAGs. At the beginning I thought maybe some quirk thing, it's too good to be true. And then it may be an artifact, they might have a strange genome, and then all the various relatives of this have the same thing. And Alejandro, it's true, no other bacteria in the world, not just alpha, has got this combination of genes. This is like the bioenergetics dream strings of genes. But of course, then we have to do some statistical analysis. But before doing that, I thought better to go back and have a look at the phylogeny of alpha proteobacteria. Everybody knows here, imagine there are alpha, beta, gamma proteobacteria, and then you have other phyla of bacteria. So somebody is working with maybe Rhizobium, these are alpha proteobacteria. But the overall taxonomy alpha proteobacteria, which is one of the largest, the second largest of the class of the proteobacteria, is complicated by a number of factors, mostly have to do with the phylogenetic analysis. And we visited together with Mac Cevallos, who is not present, but anyway, is one of our very active collaborator at CCG. We looked at the various alpha proteobacteria. This is not trivial because, you know, there have been previously proposed rickettsiales, pelagibacteriale, rhodobacteriales, and rhizobiales species have been considered before as possible ancestor of mitochondria. Whereas the rhodospirillales form a completely different set of organized groups, we call the superorder, which includes Holosporales, Acetobacterales, or Acetobacteraceae, and the new group, we call it Seric. It's a group of all exclusively aquatic, mostly marine organisms of recently introduced orders called Sneastiellales, Amcibacterales, Rhodothalassiales, Iodimonadales, and Coordimonadales. Don't ask me why they've got this strange name. They mostly come from Korean or Chinese or Japanese scientists who introduced this new species, and then the order. Seric, I hope will remain in you. The other interesting thing, the majority of this organism, of this new bacteria, relatively new versus the classical order, live what they're called in the oxygen gradient zone. They thrive in the areas in which there is quite a bit of oxygen, and then all of a sudden there is no more oxygen, like oxygen minimum zone, OMZ. Is anybody working here with oceanic environmental bacteria? I'm sure there is. No? Anyway, have you heard about OMZ? You should, because the largest OMZ in the world is just outside Acapulco. From Acapulco to the middle of the Pacific Ocean, there is the largest, the most profound, the most largest area of low oxygen in the world. And some of these guys thrive over there. So having put together this, let's just show you my first, not the last, my first phylogenetic tree. So how are they related to each other? Which are the oldest? And this, using this almost magic protein, which is a subunit of complex 1, it's a not very big protein, but it contains enough variation constrained in a certain way by which the major artifacts that arise when you put together the alpha proteobacteria and the mitochondria, and the major artifacts that can influence trees are due to the so-called the compositional bias, because the AT ratio changes twofold, etc. For rickettsia and mitochondria have a very low, and others are very big. And this kind of compositional biases, it puts in a very difficult position the rickettsiales, which are also like mitochondria. They are intracellular parasite organisms. But when you get rid of this problem, rickettsiales seem to form one of the oldest, one of the earliest branching together with some other marine bacteria. All this group, which includes Acetobacteracea, Acetobacterales, is a large variation within the Rhodospirales superorder. In the middle here, you have this large group. It contains all the seric. And then at the end, the late divergent big groups, Rhodobacterales, Rhizobiales, and Caulobacterales. These are the latest, they are the largest order, but also the latest. By the way, the majority of scientists in microbiology work with late diverging, relatively young bacteria. E. coli, for instance, is only 6 million years old, and everybody's working with E. coli. If you want to look at mitochondria and 1.8 billion years ago, you have to go very deep down here. And recently, the group of Ettema, the one who discovered the Asgard, and then other, more recently, Munoz Gomez is a nice Colombian that works in various parts of the world. They put together a work essentially, let's try this. They put together some mags found in some areas of the oceans, and they call it marine proteo-1. And when you put this kind of organisms in various combinations, here, or even just with a single protein, they branch somewhere between the magnetococcales, which some people think is alpha proteobacteria, some people think they are different, and what is called the core alpha proteobacteria. All this, the core. And basically, this marine proteome seems to be the earliest ancestors of mitochondria. So it looks a bit strange, marine proteome-1, but marine proteome-1, these groups are very important, prominent. They publish in certain journals, and they are the dominant reviewers, believe me, when you try to publish something different about alpha and mitochondria. And see here also, it's very interesting to see the distribution of the previously proposed ancestor of mitochondria, rickettsiales, HA151, Tistrella by myself and others, Rhodospirillacea, and then up here. The very first, actually, by Carl Woese himself, was agrobacterium, which is Rhizobiales, Rhodobacterial, everything. The only area in which so far there has been no, but we introduce it now, no proposal for the possible ancestor is right here in the middle, all this kind of bacteria, including the seric, which live preferentially in the oxygen gradient zone. And if you look back to the concentration of oxygen, the situation that was supposed to be 1.8 billion years ago, that would be the predominant kind of environment of the ocean. And so, this is analysis of all there are only 25 genes. 25 genes shared by mitochondria and bacteria, which define the aerobic metabolism. So cytochrome oxidase, final, cytochrome C in the middle, and the BC1 complex. And here is a statistical profile of all representative of the various lineage. I showed you before in the tree, these are in the same order, left to right, deep branching, late branching. And here is the cumulative statistical dot plot of the various aerobic traits. You see, some are more or less like mitochondria, at least 4 plus 3 different lineages, and these 4 are all seric. They seem to have an indistinguishable from mitochondria. But look at the proposal of Martin et al. In Nature. They have this marine proteo. They are so weird. They don't have cytochrome oxidase, essentially. They don't have cardiolipin or other lipid biosynthesis thing. They are really metabolically deranged. So if you think from here to here is almost impossible biochemical leap in a short time. Also the same time, so Pelagibacteriales, Rhodobacteriales, and rickettsiales, which are here, they are also statistically impossible to be close to mitochondria for the number of aerobic traits. And of course, so how do you distinguish, how do you distinguish, I got it wrong, just by the way, it's useful. By the way, in between various analyses, we recently redefine the phylogeny of Acetobacterales. Pulche. Il pulche has an Acetobacter of variation, and together with Zygmomonas, and of course, vinegar. It turns out they have a photosynthetic origin. Can you imagine to have a photosynthetic pulque? Anyway, go back here. So, to have all these groups of bacteria that statistically are not different from mitochondria is not the best possible results in the world. It helps, but you know, which one of these? Including Rhizobiales, the whole Rhizobiales. What helps is cardiolipin. And this is the collaboration with Hotto became extremely important. As I told you, I've been working with cardiolipin a long time, but I never touched, and there was a good reason about it, never touched the biogenesis. How do the cardiolipin biosynthesis is made? What's the phylogeny of it? Because it's extremely complicated, because, it's more complicated than this thing here, because there are 2 completely different enzymes that can produce cardiolipin. And what was called the bacterial kind, the Pseudomonas, is actually present in a lot of eukaryotes too. Although the other one, the so-called mitochondrial cardiolipin synthase, which has a CDP acyltransferase, has got 6 transmembrane segments. This one is just attached on the surface of the membrane, is present in a lot of bacteria, including Iodomonas. So how how can I make a tree? How can I make a rationalization from the origin of mitochondrial cardiolipin from bacteria is this kind of mess. 2 different bacteria with completely different distribution. Turns out to be that some eukaryotes got both. One we discovered is called Tecamonas, which is an apusatoa. Other, a group of Japanese before, some Bigeria, che sono SAR, they got 2. But even more interesting, one of these Bigeria has got 2, but the catalytic site here is deranged. It's not working anymore. So there are a group of of eukaryotes got 2 different cardiolipin synthetases, some of which don't work because they don't have the catalytic site. Via un'altra volta. Entonces, come si dice in español, entonces, the logic has to be that the original bacterium from which mitochondria come had both cardiolipin synthase too. That's almost impossible otherwise. And then we looked at the distribution, a relative distribution of both enzymes present, and you can see there is a real peak here at the level of aerobic monadales. Other seric don't, some rhizobiales, and that's it. Basically, only one group of the various that could have the same number of aerobic traits as mitochondria has got both cardiolipin synthases, and this becomes a very critical and very discriminatory factor. So that we can actually put together this and other independent factors, including here, for instance, aerobic traits, and see the distribution. There are only about 20 genera of alpha proteobacteria that has both cardiolipin synthases, but very few have also other factors discriminatory here. The one who has got most iodide monadales of different kind. And so from this point of view onwards, we can say from the statistical point of view, from the probabilistic point of view, the closest alpha proteobacteria to what the protomitochondria might have been are iodide monadales. People will argue, well, lateral gene transfer and so on and so on. Well, I'll leave this eventually to discussion. And then, it's going backwards? No. So this is main message in the paper we published. Aerobic ancestry of mitochondria is among extramarine alpha proteobacteria likely related to iodide monadales that lives in oxygen gradient environment. I actually can narrow it even more in oxygen gradient environments in geothermal, high temperature geothermal places. Because when they found some of the relative iodide monas, they found in a neutral island, south of Japan, which has got this thermal acidic spring, and this kind of spring we have plenty in Mexico or in Italy because are volcanic, all right? And my idea, in the ideal world, isn't it, Alejandro? Sooner or later, we're going to revisit some of the metagenomes already published by some of the colleagues in Mexico and some of these places. I'm sure there will be some of these interesting iodide monadales. Hand of the tail, 25 years, thinking how can be cardiolipin, etc. It comes here in Mexico. But then it's not the end of the tail completely. I don't know why it's not working very well, sorry. There is another story which is almost parallel. And this is, for some of you, I'm sure, you must have seen at the beginning of this year a paper in Nature showing the with high definition, the structure of one of these loci archean. Look like an enormous Golgi kind of thing with protrusion all over the place. Look at this. The body of the cell is very, very small, much smaller than alpha proteobacteria, but this, and they're all moved by actin. So, this is very interesting. I just give you an idea of science. This comes from this paper. It's a commentary in Nature by Ian Lowe, who is a director of the Institute of Molecular Biology in Cambridge. Have you heard about that place? Where Watson and Crick published polling. It used to be, when I was visiting, when I was much younger, when I was visiting that place, they were telling me there were more Nobel Prize than toilet in that particular place. So Ian Lowe is the director of this and made this comment that this protrusion gives a visual confirmation that something that is predicted but arguably the most compelling theory of eukaryogenesis called the inside-out model, made by the two Baums, father and son. This Baum here is next door in the same institute, in the same corridor as Ian Lowe. So this is a cross-fertilization you want to see. But the other thing is, when I read this paper, because all the papers I've been reading, and I've been reading a lot, believe me, about eukaryogenesis, this one here, which the most compelling, well, it's one of the least cited. It's called an autogenic model. Bill Martin, who is one of my good friend and collaborator sometimes, and he's one of the gurus of the, says it just it's crap. Excuse me, it's not very interesting. However, and I looked at it, I read it carefully, and in fact, some of the things, yeah, but then he relied to a very important concept. This kind of protrusion here, they need like in a Golgi or in mammalian. Anybody working with membrane traffic here, mammalian cells? Phosphodiethyl inositol, phosphorylate PIP, they need that particular type of membrane lipid, phosphorylated and not. And look what it says. This Robert Mitchell, another Englishman at another university, and as expert in inositolipids from an archean origin. Archean origin? But all the other lipids we have been looking with the auto, they're all coming from the alpha proteobacterium. Well, come on, how come? From archean origin, just from previous data and no phylogenetic tree. Well, I looked into it. It's going the other way around. Disculpa, eh? This is, you don't need to be too smart to be a scientist, but Omar, che passa? Fammi venire qua. Anyway, disculpa, arrivo, eh? No. I lost my practice here with this. Actually, questa slide. I want this slide. So guys, by myself, with my little trees, I looked into it, and after 3 rounds or revision, because nowadays even Fab's letters, 3 rounds of revision, I found out that Mitchell is completely wrong. Consequently, the inside out, the most compelling, is also completely wrong. Because inositolipids, guess what? They come from alpha proteobacteria, 2. So this almost in parallel, I got these two little publications, eukaryogenesis. But now, there's another thing I want to talk to you about. So basically the science is over. But this is called altmetrics. We were discussing today altmetrics. This little number in this little circle is 310. Anybody is familiar with altmetrics? It's essentially a metrics by this company called altmetrics, which measure what is called the online attention, the internet impact, the wave, the size of the wave of interest that your paper generates in social media. 310 means there have been 8,700 downloads of this paper in 3 months. There have been 20 media outlets that made articles, and there is even one video in YouTube by some scientist that actually explains with the video our paper. Thank you very much. 310. To give an idea, my most prestigious recent paper, I got 17. Is anybody here with an altmetrics above 500? Have you ever checked your latest paper? Check it, because one great satisfaction is this is altmetrics of one of those papers of my competitors, my reviewers, very nasty reviewers, published in Nature Evo, 85. In 2 years, no, almost 2 years, it has got 3 Wikipedia page, but that's. So, this is one of the satisfaction at the end of a career, or almost. When you get a project, you get a certain answer. Maybe it's not the final answer. I don't know. Let's see. But it's a very good answer. And according to the media, well, it's more popular. Okay, we shall see the science later on. And I wish you the best altmetrics you can possibly have. And with this, I will try to finish with thanking all the colleagues, in particular President Otto Geiger and the various other colleagues have been collaborating with here in Mexico. I skip others, including some Japanese. They wrote to me, some Japanese, because they say, so basically I'm actually on the process of analyzing some more of these Japanese bacterial things, and with this I thank you very much for your attention.
Muchas gracias, Mauro. Vamos a las preguntas y empezamos. Bueno, tenemos tiempo para preguntas. Aquí.
Very nice talk. I wonder whether structural biology could be used to find similarity between a large collection of proteins and then establish a stronger relationship between some bacteria and mitochondria.
No, very good question. If I can go back. Well, when it comes to those respiratory chain systems, we have detailed three-dimensional structures, mostly from cryo-electron microscopy. So all these are structures, are very detailed. And the other thing then it also becomes very interesting. In the structure, for instance, of tetrahymena, with the first protist for which we have mitochondrial respiratory chain, there are subunits which are normally not present in any other eukaryotes, which are related to SIRF1, which is normally nuclear encoded protein. So this kind of information has already helped. Of course, nobody has ever looked at the more, let's say, red algae or some other very early branch. Tetraemena is a ciliate, it's a very kind of tangent, has got its own very high diverging rate of mutation, but in principle, yes. With cryo-electron microscopy in particular, it could be. This kind of panorama here is not complete. I'm sure it will be, well, it will be implemented further by filling
But
in.
you are considering only presence and absence of certain subunit. And I was thinking more in the,
In the statistical analysis, you mean?
yeah. That's right.
Generally, yes, but no, with a bit of a flavor, because, for instance, we give a weight in this particular, it's absence and presence, but we give a weight for synteny, because there are some synthetic combinations which remain in mitochondrial DNA. We give a certain cap, for instance, for certain. There are multiple versions of one or two of these genes, and in eukaryotes there is only one in bacteria. So it is a modified standard 0-1 binary absence and presence, so we give away. But nevertheless, even if you take away that particular, this kind of peak here remains.
¿Alguien más? ¿Dónde está?
Aquí, este señor.
Aquí. Okay.
Very interesting. Thank you. What is advantage having two cardiolipin synthesis?
Otto, would you like to, he has got a very good answer. But I can, you want to do it?
The reactions they're catalyzing, they're different. So one is essentially like a phospholipase D and leads to an equilibrium which is not very much on the side of cardiolipin. And the other uses highly energetic compounds like CDP diacylglycerol and really has the ability, when it gets to equilibrium, you have 20% or more of cardiolipin, and this is what you see in mitochondria. Many bacteria have only about 5% of cardiolipin.
So basically,
because they catalyze different reactions, well, the equilibrium is more on the side of cardiolipin or not so much.
So you can also presume that the presence of both, it gives more flexibility for the bacterium, because cardiolipin is also important for the structure, to help the structure of the respiratory complexes. So when you have enough phosphate, then it's easy to use the PLD. When there's not enough phosphate, and you have a little bit of phosphatidylglycerol, which is the precursor, like in mitochondria or like in oligotrophic environment, it's certainly favored, particularly in most marine environment, it's much favored the mitochondrial type, the one which is driven by energy release.
Yeah, and maybe an additional comment there. So if you use this pathway where you can make a lot of cardiolipin, this is more costly. Yeah, essentially you have CDP diacylglyceride as a precursor, which to make it you need ATP. In the other case, it's for free.
The alt matrix. Everybody knows the alt matrix or no?
The mitochondria uses a particular genetic code.
Yes.
Has anyone explored whether these bacteria or some members of
They have the same normal code. There are lots of theory how the genetic code of mitochondria came about, and there are some variations in some protists, even for the original code. There is no possible way you can connect for as far as I know. at the level of the code. And so this, I wouldn't say a mystery, but something I don't have any knowledge, but it's not very much followed in the literature of origin of mitochondria, this particular concept.
Thank you.
¿Alguien más? ¿Alguna otra pregunta? Bueno, pues...
Okay, final one.
Yeah, very exciting and excellent work, Mauro. Thank you so much. I am wondering what's next. I mean, do you have, have thought on another evidence or another approach to, I know this is conclusive evidence.
Well,
another one?
Well,
I mean, what's next in this work?
As we actually, I haven't shown you, but there are some media outlets that maybe you like to have a look at. What is it? No. It's here. If you want to have a pre-digested or a way of this particular paper, you can look one of these, in particular, the second one, which is in a science website, which is articles describing the story, even and they ask exactly the same questions as journalists when they interview us. Two things easy, which I already mentioned. First, extract more information from the Japanese colleagues, possibly in a way. There are at least 3 different new mags. They already told me. They look very much more interesting than the one we already analyzed. I hope so is the same. That would be. The other thing in the future, if we can actually look with some specific probes to look for some Mexican iodative monadales, why there is no reason why they shouldn't be here. They found they found them in India, in an Indian bioreactor. So they are more spread than we normally know. A third, which we haven't really touched here, but the biomedical implication. The biomedical implication that cardiolipin is made with 2 different enzymes which have a different distribution in eukaryotes is very much useful to understand the evolution of mitophagy. Because cardiolipin is the first flag you can think of, something which is not of the rest of the combination of the original eukaryotic thing. It's the first flag. If something goes wrong in the mitochondria, you as a new cell, you don't want to have mitochondria or ex-bacteria and slave that become mitochondria. They don't work. You need to have a quality control system. When they don't work, you want to get rid of them. You want to eat them out. And the origin of mitophagy must have been exposure of cardiolipin on the surface, like kind of a flag. From that to the mechanism we know now, there are 16 different flags for mitophagy in eukaryotic cells of mammalian. And I'm studying now, in collaboration with Bill Martin, what was in between. So what happens with plants, with red algae and other things? How do they cope when the mitochondria don't work well? So this, I think, for me, is fascinating, considering all what the work I've done before in cell death signaling and cardiolipin, or even have a fluorescent cardiolipin to play with, etc. So that's, I think, is a more fascinating long-term, project that comes from this work.
Thank you.
By the way, I have a review on the journal Autophagy, talking exactly about this and to lay down the project. It's not accepted yet, but possibly soon.
Pues muchas gracias, Mauro. Te agradecemos nuevamente tu participación y bueno, gracias.